All right, good afternoon, and thank you for your patience as everyone joins us in the webinar room. We will begin in just a moment. All right, welcome to our MoIP webinar, Understanding Plant Invasions Using a Traits-Based Framework with Dr. Gabriela Nunez-Mir. My name is Amanda Lands-Ramrup, and I am the Events and Communications Coordinator for the Missouri Prairie Foundation and Grow Native program. Thank you all for joining us today for the first Missouri Invasive Plant Council webinar of our 2025 series. Now for a few Zoom basics before we continue. When you have questions during the presentation, please put them in the Q&A section on your screen, which you will find at the bar at the bottom of your Zoom window. Questions placed into the chat will be asked to be redirected to the Q&A section. Ellie Germeroff, our MPF Conservation Program Associate, will join Dr. Nunez-Mir on screen at the end of the presentation to read your submitted questions aloud. Everyone streaming in has our gratitude in advance for your thoughtful live submissions. Now I'd like to more completely introduce Dr. Gabriela Nunez-Mir. Gabby is an assistant professor in the Department of Biological Sciences at the University of Illinois at Chicago. As a quantitative ecologist, her research focuses on leveraging the wealth of knowledge held within large bodies of ecological data to investigate the dynamics of biological invasions and other subjects of conservation concern. The innovative statistical techniques she employs, which include machine learning, Bayesian, and geospatial methods, have allowed her to produce findings that are uniquely useful and broadly applicable across geographic and taxonomic contexts. She obtained her PhD from the Department of Forestry and Natural Resources at Purdue University in 2018. Prior to joining the faculty of UIC in 2022, Gabby was a postdoctoral fellow at Virginia Commonwealth University, studying the range dynamics of spongy moth. Gabby, thank you so much for taking time out of your schedule to join us for a webinar on your research. I'll now turn things over to you.
Thank you so much for having me. And thanks everyone who is on this Zoom right now. I cannot see you, but I feel you in spirit. Thank you for being here. And also thank you for that really nice introduction. I was going to talk a little bit about what I do, but that makes my life easier. So I am an assistant professor over at UIC and I am a quantitative ecologist, which means that today's talk might be a little bit different if you're more used to being outdoors. In fact, I always refer to myself as an indoorsy ecologist. And in the lab, we study biological invasions. So that's insects such as the spongy moth on plants. Today, we will be focusing on the research that we do on plants. But the whole unifying theme is that we use data to understand how these invasions operate at very large scales. So we're talking regional to continental. Lots of times is, okay, what's going on across the United States, for example? And with that, we think a lot about how our research, even though if it's at a very large spatial scale, how can our findings help management and the work that is happening on the ground? So keep that in mind as we talk. I'll give some notes here and there of how our research will be used for management purposes. But if you have any ideas, please reach out. We're always excited to do that kind of work because there's nothing more rewarding than seeing the research jump out of the page and become useful in real world applications. A real world context. So as I said, we do plants, we do insects, all sorts of topics in the lab. But today I will be focusing specifically on our work understanding and forecasting at times the dynamics of invasive species, particularly plants, through their species traits. So with that, let's just start with why traits and what are traits, right? So species traits are any measurable characteristic of an organism that tells us something about its biology or its ecology. Now, these characteristics, which can include things like flowers and the height and leaf characteristics and stuff going on with the roots or life history or characteristics of its range. All of these things, all of these measurable factors tell us a lot of things about these organisms. First, it tells us about their evolutionary history and potential, it tells us a lot about their life history and their strategies and their place within the ecosystem. So, a lot of times, these topics in ecology are very, very, very hard, sometimes impossible to quantify directly because they're quite complex. So, using traits as proxies for these mechanisms, these details that I have described of these organisms that can't be quantified directly, we can use the traits as kind of a indirect way to study them, and that's why it's such a powerful framework. And we're very excited to use it and see how many fun findings we can find using or we can derive using that framework. So speaking about these fun findings, during my PhD, actually, the work, and this is how I got started with species traits, we were able to create a model just using 6 traits that was able to identify invasive non-native woody species. Out of all of the non-native woody species, we had a list of about 900 different species in the United States. And our model had an 86% overall accuracy in distinguishing out of these non-native species which ones are invasive, which ones are non-invasive. And those characteristics were asexual reproduction, dispersal methods. If they had a long distance dispersal method, that was made them more likely to be invasive, but also primary growth form. So with that, we're like, okay, so this is useful. We can use that just not only to understand the mechanisms, again, asexual reproduction, long distance dispersal, what is actually leading to invasion success, but then this is also information that can be used in risk assessments. Oh, does the species have this characteristic? Maybe we should prioritize it, especially in programs like EDRR programs, early detection and rapid response. But during this project, again, I did this 2019 is the date on the paper, a while ago through my PhD. And one of my collaborators was a scientist over in Europe. And we had this huge discussion about whether some of those species on our list should have been classified as invasive or versus non-invasive. And we were using the federal classification from the Forest Service for that. But that European scientist had a different perspective. And that got me to think, okay, so what really defines invasion success? What makes a species invasive other than this rules that we have, like in the federal law, is encoded in a very specific way that's supposed to be a species that is non-native to an area, that has been established, spreads, and causes substantial ecological harm or economic or damages or harm to human health, right? So it has a definition in law, but that is a human definition, right? It's not necessarily an ecological definition. So it got me thinking a lot about that. And right here, you have 2 pictures of 2 invasive species. On the right, you have common reed, and on the left you have Australian pine. And if I show you these 2 pictures, you're like, yeah, then I tell you they're invasive. You're like, yeah, that makes sense. I mean, they are dominating the picture, they are forming monocultures. But if I show you these pictures or these maps, the story seems a little bit different, right? So common reed is all over the place in the country, whereas Australian pine has a much more restricted invasive range. So it's not found everywhere in the United States. And that begs the question, okay, are these 2 invaders the same? Are they equally successful? So while I'm thinking about this, a paper actually came out a few years later in 2021, where they disambiguated invasion success into different dimensions. So they had that a species could be an invader that is very successful locally, so very abundant, or it might be a generalist invasive species that is able to thrive in different types of habitats and environmental contexts. Or perhaps the species is found everywhere in the continent, so it's very widespread, has a very large invasive range. Species that could be considered super invaders would be successful across these three dimensions, but there are many cases where they are unsuccessful just in one or just in two. But it brings back that definition of invasion success into an ecological definition or an ecological context, where instead of having a social anthropogenic definition, right? So just to kind of really establish this idea, let's compare these two species. So we've got tree of heaven, which is abundant, a generalist, and widespread, right? It's found in over 2,000 counties across the United States. It was introduced in the late 1700s. But then we have another invader like Suriname cherry, which is also abundant, also generalist, so successful in two dimensions, but it has, as you can see in the map, quite a restricted invasive range as well. It's found in about 26 counties, at least at the moment that I got this information. And it was introduced in the early 1900s. So it's had quite a bit of time to grow in its invasive, in its range and spread across the continent, and yet it hasn't. So that really made us think, can we find the reasons why are some of these invaders more successful than others in these different dimensions? So we proposed this research that is now funded by an Biota Award. We were able to secure that by proposing this research. And we decided, okay, let's test different mechanisms by which a species, an invasive species, could become successful in that widespread particularly in that one dimension, in the invasive range size one. So we thought things like how broad are their their environmental tolerances, so their niche, how aggressive they are, maybe that has to do with it. Maybe it has to do with adaptation ability, or maybe it has to do with self-reproduction and dispersal. As you saw before, invasive non-native woody species were separated from the non-invasive woody species by the fact that they were able to reproduce asexually. So we thought of all these mechanisms and then the traits that we could use as proxies to indirectly test the role of each of those mechanisms on success in these different dimensions, specifically the one of becoming more widespread at the macro scale. So since 2021, when I got to the University of Illinois at Chicago, I and a fantastic team of undergraduate researchers have been putting together this huge traits data set for invasive plant species in the United States. And this is almost out many years later. It's been a lot of work. We have traits data for, I believe, over 20 different species traits, and that's for over 1,000 invasive plant species in the United States. And this data has been gathered from all of these sources that are readily available online, but not in a way that can be used for data analysis. So we're talking about fact sheets. I'm sure many of you have seen them or produced them yourselves. So we went to sources such as CABI Compendium. We visited North Carolina State's Extension Plant Toolbox, the Gypsum Herbarium data set, USFS, the Forest Service Fire Effects Information System, University of Michigan Climber. So everywhere, actually, the Missouri, I can't believe University of Missouri Herbarium as well has fantastic fact sheets, which we used as well. So yeah, just going all throughout the internet, reading these narrative descriptions of the species and manually inputting all that data into a spreadsheet, which will be published for public use shortly, hopefully within the semester. So stay tuned because that will be out there for everyone who's interested if you'd like to have access to this data. So using that data, we set out to investigate, okay, what sets apart which of these mechanisms that are associated with traits set apart species that are successful at neighborhood, macro scale, or landscape scales. So, in other words, are they abundant? Are they widespread? Are they generalists? In conversations with other scientists, I found out that Dr. Bethany Bradley over at UMass Amherst was working on a similar project and said they were classifying invasive species and other non-natives that are here already in the United States into these categories. So we decided, okay, we'll bring our traits data, you bring your categorization data, and we are going to see what's going on here. Now, for today, I will be showing some preliminary results just on that macro scale widespread dimension, as I mentioned. As you saw before, my lab is called the Macroscale Ecology and Conservation Lab. So it's the dimension I was most interested in. So we started there, and these are the results that I'll be showing shortly. So we found with a subset of the data that niche breadth may be the mechanism by which these species are becoming widespread. So they're becoming established and spreading across the continent. So becoming successful at the macro scale. That's among all those other mechanisms that I had mentioned before. More specifically, when we dug in a little bit more into the data, we found that hardiness zone range, so how many different hardiness zones can this species be found in, was positively related with the number of counties in which each of these species was found in. So the broader their hardiness zone range, the more likely they were to be established in more counties, so have a much larger, wider invasive range size within the United States. And that this preliminary result came from data from 275 species. And we found actually that hardiness zone range was able to explain 15% of the variation of the number of counties among those species. That's a pretty decent number in ecology, at least. Now, we also found that elevation range played a role. And now this is telling kind of a story about temperature, and that the broader the temperature tolerances of these invasive plants, the more likely they are to be found across the United States. And now that might not make or it might make a lot of sense now saying it, but we've got data to back it up. If these species are have temperature tolerances that are quite broad, and elevation range can kind of say that, because you go up in altitude, you're getting a different type of weather, right? It's getting colder. And this variable on its own elevation range was able to explain 9% of that variation in the county. So we're really getting onto something here. Now, the purpose of this project more broadly is to be able to identify out of these species the ones that have been recently introduced, not the ones that we know, the enemies that you know, right? From these newcomers, these recently introduced non-native plants into the United States, which one of those are going to become a problem in 50, 100 years? Right? So basically, we're trying to find America's next top invader. Which ones are going to be those super invaders? So I'll, as I said before, I'm always really interested in see what's going to happen. Very excited. So I went through the data with the preliminary results that we have to pull out some species that could be candidates once we finish our project. Could be one of those America's next top invader. And one that showed up was actually Japanese chaff flower. Now, I believe this species is mostly in Kentucky right now. It might be in Missouri as well. It's coming up to Illinois, from what I hear from my friends with boots on the ground. So, Japanese chaff flower, Acaranthus japonica, has been in the, at least as far as we know, in the U.S. for 38 years now. In the elevation range is quite broad. Hardiness zone range as well. And just going by those preliminary results, this is what we pulled out. Now, It was really fun. Last time I gave this talk at a conference at ESA, actually a couple of years ago, someone came up to me, a practitioner in Kentucky. He's like, "Wow, it's crazy that you're talking about Japanese safflower because we're dealing with it right now, and it's so awful." And talking to another colleague over at the University of Illinois Extension, he's like, "Yeah, I grew up with that stuff. It's pretty bad. Yeah, it's really cool. So something to keep an eye out for." Japanese safflower. When your results, us that are dealing with data in front of a computer all day, honestly, again, indoor ecologists, we're not going outside. And the folks who are outside and us from the data perspective, when those two approaches to understanding invasions come together, it's really, really cool to see. Now, that is one aspect and one thing that we can do. We talk a lot about forecasting invasiveness, what's coming next, what's getting in, and all of that. But that got me thinking, what else can we do with this traits framework? I mean, I said that at the beginning of the talk, always thinking, what else can we do with this data now that we have it, right? And the next logical progression from understanding establishment and spread would be impacts, right? That's the part that makes an invasive species an invasive species. It's that we notice it because it's bad. It's doing something that we don't want it to do, either to our ecological systems or economic property or whatever. So we started thinking, okay, can we use this traits framework to understand invasive impacts? And we turned our eyes below ground for this work. By the way, the work that I'm going to be talking about now is actually what brought me to you. The good folks over at the Missouri Prairie Foundation and the Invasive Plant Council, they read this work. So I'm really, really excited to show it to you guys because it's more complete than what I just talked about. So, all right. We thought, okay, can we use traits to understand the impacts of invasive plants on soil microbial communities? So as I said, we are turning our eyes below ground now. And part of the reason why we wanted to look below ground was because, okay, we know that invasive plants can affect soil microbial communities through, well, whenever they have exudates, for example, if they're allelopathic and those are antifungal components, or perhaps we know also that through leaf litter, that the chemistry of leaves can change that leaf litter and therefore have a cascading effect on soil microbial communities. However, in the literature as a whole, we still have conflicting findings. It's not very well established the ways and the effects that invasive plants can have on soil microbes. And part of that is because, yes, we know what the leaves are doing or have an idea, and we know about allelopathy, but we haven't really looked at traits of roots as closely beyond, as I said, allelopathy or nitrogen fixation. That's another one that has an effect on microbial communities. And that's because, in part, root traits are very hard to measure because you have to pull the whole root system out. And if you've ever done that kind of work, you know exactly what I'm talking about. So part of why we don't know is because root traits, below ground traits, so characteristics of the roots themselves have not been studied as closely. And because they're very hard to collect and they can be very different morphologically, so in their shape, in their structure, in their function, in their chemistry. And invasive roots may change soils through different ways, through bioturbation, through exudates, that's chemicals that the roots are exuding, so expelling into the soil, and through the recruitment of symbionts. So because there are all these ways that roots can affect, my co-author in this work, Dr. Matt McCary over at Rice University, and I set out to collect data on the microbial composition, plant species and their cover, for 377 plots across the United States, the continental United States. Oh, actually, we had some in Hawaii as well. So across the entire United States. From NEON, NSF NEON. Now, NEON is a network, for those who are not familiar, from the National Science Foundation, a network of sites that are all over the country, and they collect all sorts of data. It makes my job a lot easier. They're really fantastic, and I'm very lucky to be a scientist while the network is ongoing. So from NEON, we got data for 1,800, over 1,800 native plants and about 187 invasive plant species that were within these plots, these 377 plots across the United States, as I said. And for those species, we set out to obtain root trait data. Now, with root traits, I mean everything from root diameter to root tissue density, so morphological characteristics of roots, but also some chemical characteristics of roots. We've got root carbon concentration, root nitrogen concentration, and also the ecological characteristics of roots. For example, mycorrhizal colonization intensity. So as I said before, sometimes these roots are used to recruit symbiotic organisms. And we got that data from these fantastic databases that are available right now. That's Fred, Groot, and Try Plant Trait database. So we set out to collect all of that. And if any of you have done some traits research before, you will find out and you will know that it's really hard to compile characteristics from every species. So we had to do a little bit of what we call data imputation. And it's just an advanced statistical method and phylogenetic method that takes a lot of information that we've already provided, including evolutionary information of the species, phylogenetic information, to plug in some of that data that we were missing. So our final data said we did have to remove some species. We ended up only having 693 native plants and 94 invasive plants. Now, with all this fantastic data, we set out to answer 3 questions. The first one being, are the roots of invasive and native plants different? Because if they're not different, it's likely that invasives won't have a distinct effect on soil microbes, at least not through the roots, right? So we set out to answer that question first. The second question, are the microbial communities of invaded and uninvaded plots different? So, okay, we'll see if the roots are different. Now let's see if the microbial communities of plots with invasive and without invasives are different. That was step 2. Step 3, if we do find differences, are these differences in microbial communities attributed to the roots, right? Because as I said before, leaves can be causing effects on microbial communities, as well as other characteristics or other traits such as nitrogen fixation and allelopathy. So we just wanted to see, okay, these differences, are they actually due to these other traits that we're studying? So for the first question, we performed a suite of phylogenetic regression models. Basically, it's a kind of regression model or a kind of comparison between 2 groups that takes into account that species in general, plant species, are not completely independent from each other. Species are related to each other. You know, there are species that belong to the same family and the same genus. So we need to take that into account with when you do analysis with such a large number of species like this. So we had whether they were invasive and whether they were native, and we compared all those 8 traits that I showed you earlier, 9 traits, if they were different from each other. And we found that invasive plants actually had higher specific root lengths than native plants and lower root tissue densities. Now, that is kind of telling us that these invasive roots tend to be really cheaply made and they grow fast and they're just going out there, you know, trying to scavenge for resources. So that's kind of the strategy that invasive plants are taking, more so than our native plants. All right, so that was question 1. We found that they are indeed different. Now, question 2 was, are the microbial communities of invaded versus uninvaded plots different? Now, we took our 377 plots, which I must say, if you add these up, it doesn't add up to 377. And that's because some of these plots were sampled more than once, some of them even 3 times through time. So we used each of those separately, obviously taking into account that it's the same plot statistically. So we separated those into 3 groups. We have our highly invaded plots, which had an invasive cover of over 50%, moderately invaded plots, which had invasive species in them, but were anywhere from more than 0%, so like 0.5 to less than 50. So that group is quite broad when it comes to invasion level. And then our plots that were not invaded, so native, all native plots. And you can see the numbers of how many plots of each we had. Thankfully, we didn't have that many highly invaded plots, right? Maybe not so good for the investigating part, but it's nice to see in real life, even though the moderately invaded, we did have quite a few of those. All right, so to study the differences in microbial communities among those three groups, we compared the microbial composition of these plots by two ways. First, we did an equal multivariate dispersion test, and basically what that's telling us is, are the plots within this one group, let's say the native group, are all the native plots more similar or more different to each other than the plots that were categorized as highly invasive? That's what that test does. And that first test is necessary to do the second test, a Permanova, which what it does is it tells us, okay, out of all those microbes in the soil, how different are these groups from each other? We were not able actually to run that second test because the first test failed, which sounds bad, but actually tells us a lot. And I'll tell you what it tells us in a little bit. So what we learned from that failed first test is that it failed because the microbial communities of invaded plots were a lot more similar to each other, so a lot more homogeneous than those in uninvaded plots. So if you turn your eyes to this 3D diagram, you can see that the blue dots, which are those native, uninvaded plots, are all over the place, which means that the communities, the microbial communities within these plots are very different from each other. But you can't even barely see, you can't see the red ones, right? They're all clustered in one spot. And again, on the other hand, what that means is that those plots were very similar to each other in microbial, what we call like microbial composition space. So their microbes, the communities of microbes in these plots were very similar from plot to plot. And one of those plots could have been in Alaska, and the other one in Missouri, and the other one in Florida, and the other one in Massachusetts. And even though they're in very different geographical and environmental contexts, which by the way, we had all sorts of habitats. We had forests, we had grasslands, we had even a couple of croplands in there. We found that their microbial communities were very similar just by way of being highly invaded. Now, the moderately invaded is a little bit of a different story. They still were found to be very clustered, a lot more so than the non-invaded ones. But you can see that there's a little bit going up. And again, as I said, there's a lot of variation of how many invasive plants are in those plots. So we weren't that surprised. But that result that you see, that B graph over here, it tells us that when you compare the level of dispersion, so how different non-invaded are to highly invaded, yes, they're very different. That line and whiskers, the dot and whiskers, is very far away from that zero dotted line. When you compare non-invaded to moderately invaded, that difference gets closer to the zero, but it's still significantly different. But when you compare, moderately invaded to highly invaded, no difference in dispersion among those two. All right, so that was question 2. So we found so far that invasive roots are different than native roots. So roots of invasive plants are different than the roots of native plants. And we found that invaded plots tend to be more homogeneous in their microbial communities than native plots. Now, third question remains. Are these differences that we are seeing, can they be attributed to the roots, or are they something else, caused by something else? So we approached this question in 2 ways. First, we did some variation partitioning to differentiate the relative effects of native and invasive plant root traits in how they structure the microbial community when they're in the same plot. So we're looking at the roots of invasives and of natives in the same plot and seeing how much one affects or how much each of those affect the microbial communities. And the second approach that we used was a longitudinal analysis. So as I said before, some of these plots were actually sampled in several years. So we did have, for some of them, plots that were sampled in year 1 and year 2. And we used those plots, that difference from 1 year to the next, to see, okay, if this plot changed its microbial composition from 1 year to the next, and it also changed its invasive plant cover from 1 year to the next, are those changes in the roots from those invasive plants, can they explain that change in microbial community? All right, so we found actually that invasive plants explained almost twice the variation in microbial composition than native plant roots. So invasive plant roots explained about 27% of the variation, whereas native explained about 12%. Altogether, though, they explained quite a bit of microbial composition, which highlights, it emphasizes the role of plant roots in structuring soil communities. And we believe they have been overlooked for too long. We should be putting more emphasis in getting more data on the traits of roots of both native and invasive plants. Now, the results on the right are from the longitudinal study. And all you need to see here is that CN ratio, this first root trait over here, was able to explain 20% of a change in microbial composition from 1 year to the next. That's just one trait of the roots, CN ratio. We also found that specific root length, which we saw in question 1, where it was the difference between invasives and natives, was able to explain about 6% of the difference between in the soil microbial communities from 1 year to the next in the same plot. So to recap what we found here for question 1, invasive and native plants differ in specific root length and root tissue density. For question 2, microbial communities of invaded plots tend to be a lot more homogeneous than those that are in uninvaded plots. And for question 3, invasive root traits do indeed contribute to these observed differences in microbial composition. Now, the implications of these findings is that the root traits that we saw that were more likely to be associated with invaders, being, the high CN ratios and the high specific root length and low root tissue density, it just means that they are putting out faster root growth for foraging, and that is modifying the soil in ways that affects the growth of soil microbes. Beyond that, these invasive plants are altering soil communities in very similar ways. So this is producing very homogeneous, very similar microbial communities, regardless of the ecosystem type and regardless of the geographical context of that community. And this is very important because such homogenized microbial communities may result in the loss of very important functions, community, ecosystem services that are occurring at larger scales, perhaps at regional scales. Something such as decreased resilience to disturbance, for example, which is a service that occurs at a larger scale, or the loss of microbial mutualists and beta diversity. And with that, I'd like to thank you all for being here and for bearing with the technical issues. And a very special thanks to all of my collaborators and co-authors here at UIC, at UMass Amherst, at the Forest Service, at Rice, the wonderful people who put together these databases. I get to have this very fun job thanks to you. You go out there in the field, get the data, and then I get to play with it, and that's fantastic. Thank you so much. And our funding source, the Walter Foundation Biota Award. Thank you all.
Great. Thank you so much, Dr. Nunez-Mir. That was great and really fascinating. It's awesome to think about these problems that we're facing on this larger, sort of broader trait-based scale. So really awesome. And I feel like we're all thinking about soil. So this was really interesting for all of us, I'm sure. Yeah, we have some great Q&A questions. So I'm just going to read them off and we can go from there.
Awesome.
So one of our questions was in the beginning of your talk, you were talking about predicting whether a plant or, who America's Next top invader, which I thought was really funny. They were asking about false positives or false negatives. So were there any that you would predict to be highly invasive, but haven't seen that, or vice versa? Do you have thoughts there?
I actually do. That's a fantastic question. So, we haven't completed that project yet. So, when I pulled out Japanese chaff flower was just off of the preliminary findings. But can everyone still see the slides? I want to show something.
Yes. Yep.
Great. When it comes to those false positives, we had those issues in my PhD work. And if I can show you, okay, this is the graph. So as you can see here, we have an organization in which it seems like invasives and non-invasives are mostly separating, but there's still some spillage from the non-invasives to the invasives, which would be a false negative, right? And that's what kind of got us thinking maybe some of these species are actually invasive or have the potential to become invasive, but we don't currently classify them that way. And there are a couple of red points that are actually also bleeding into the blue, which might mean this might be invasives that are only successful in very specific dimensions. So maybe they are highly abundant in a very restricted range or in a very specific habitat. So that is a fantastic question that I would love to have an answer once we finish the project and time goes by and I can actually see, okay, we were right with these, we were wrong with those. But in a case like with predicting or forecasting invasive species, it's hard. I was going to say it's better to have false positives just for, you don't want to let something go by. But the reality is that we are constrained in resources. So it's a problem both ways. That's a great question.
Hmm. Definitely. Thank you so much. That's so interesting to consider. Yeah, we're predicting years out potentially. So yeah,
Yeah.
this is awesome work. We had one question about you know, how could how could knowing these traits their like the elevation which they grow and their temperature ranges, how exactly do you say that this helps with eradication of the species? Um, yeah, if you could just speak more on that.
Yeah, thank you so much, whomever provided this question, because I actually forgot to mention our plans when it comes to management applications of this work. All right, so we are planning on coming up with a list, as I said, of those top invaders. But beyond that, we're actually also planning on putting together brochures with information about these species so that, and that's what I meant with early detection and rapid response. If the species is not yet in your state, we hope to inform folks on the ground of what to look out for, what could be coming your way. Now, that's one side of the coin once we've identified which of the species those are. I use that trait information to do that. Now, beyond that, folks that read these findings could use those traits in their own risk assessment protocols if they choose so. And with that traits database that we're putting together for that includes many recent introductions, that also facilitates getting that kind of data on those specific traits. So if you choose to include that into your risk assessment protocol, the data is already there. It's easy to find. So I know how long those assessments take. I or not participate, but I sit in the meetings for the Illinois Invasive Species Council, and I hear how this stuff gets on the list, and they're really long. So we're hoping to get data out there, quantitative data, and make it more accessible for everyone.
Awesome, thank you. We whenever you were sharing about the publication you were working on with your team of students that's helping you with looking at all the fact sheets, we had questions saying, how will we access this? This would be really great compiled information. Can you speak to that if that will be part of the publication?
Well, the data set and the description of how we collected this data or the methodologies of how we collected them will be published. I hope soon. It's one of those things you keep
Mhm.
saying, yeah, by the end of the year. But definitely by the end of the spring, that publication will be at least under review. And I don't know how to reach out to everyone and tell them like, hey, it's published. We're submitting to ecology. So maybe keep an eye out for ecology data papers because the whole spreadsheet with, we're talking environmental tolerances. We have maximum height for the species. By the way, I must say that these are all species level averages for each of these traits. Well, those who work with fact sheets know that that's the best that we can do. We've got also dispersal methods. We've got introduction vectors. We've got at least what we think is the introduction year into the United States. So a lot of good data. So stay tuned is all I can say. Oh, actually, you know what? How you can stay tuned? My website is on the last slide. So just keep checking in with us, or at least by the end of the spring, and see if that publication is up already.
Definitely. And we will, us at the Missouri Prairie Foundation are going to keep an eye, just in general for your really useful and your useful contributions to the field. But yeah, we'll keep an eye on that. But that's a really, of course, very useful use of everyone's time. We now so I'll shift a little bit to some root questions. So we had some people that were more curious about root structure and invasiveness. Can you maybe speak to the difference of root structure in invasive species? And if you would if you would like to speak more on this sort of root structure in invasive species in the future. Is this something that you're going to be working on more?
Yeah, certainly. So let me start from the end and back to the beginning. So this is very initial work on how root traits very specifically affect the soil microbial communities. As I said before, we know how leaves do it. We know about nitrogen fixation, all of this. So this was very like first in its, I don't want to say first in its kind, but it's a beginning of that line of research. So it was quite broad in the way, as you can see, or as I said, we use species level averages for this, which doesn't maybe give us that localized information about how a specific community and a specific ecosystem is working, what are the mechanisms that lead to these patterns. More so, we describe the patterns that are happening across a very large scale. So the next steps in our work, my co-author and I, we will be looking at now individual level traits, also looking at characterizing those microbial communities a lot more at a much higher resolution. We use something called PLFAs, which can be quite broad in the way that you describe a microbial community. So we started big, and we're trying, or with the future work that we're going to do, is to get more localized, more specific, and to see what are the actual mechanisms and processes that are happening. Now, to talk more broadly about invasive plants under roots, one thing that generally makes invaders so successful in establishment and in spread is that, so becoming very locally abundant, is that they can be very good competitors. And I think that that's the evidence, what we saw, our findings are evidence to that. So we saw that these were roots, the fact that they were lower root tissue density and that they had longer specific root lengths. It just means that these are roots that are made very cheaply, as I said before. So not expending a ton of resources into this, but still growing very fast and very long. And that improves foraging ability, which means that these plants then are a lot better than others at getting any resources that are available, whether they're plentiful or not. So that makes them better competitors, and they can then beat out our native plants in that search of resources. And to give you kind of the picture of what happens after, so now they're better competitors, they're taking more resources. That means they also have more fuel, more food for reproduction. So they're putting out more seeds, and it's just a vicious cycle, right?
Definitely. Yeah, thank you. Very well said. I think we've all seen that sort of cycle of once there's enough invasives in an area, it just tips over. And we have a question about potentially, is there a possibility, because there is such a difference in invasive plant root systems and native plant root systems, is there a possibility of introducing a virus or a microbe that would control that? Can we use something special against these invasives? Have you heard anything about that?
Listen, I'm laughing because I feel that every time the idea of like, let's bring something else,
Yeah.
something like those stories a lot of times go wrong, but sometimes they do work out, right? I don't know for sure. And these, as I said, these are very, very new findings that we've just put out there to the scientific community. That's a great question that I don't think I have a good answer for, frankly. That's really
Yeah.
interesting.
Speculation, and we'll all be wondering that, and we'll keep our eye on what happens there. But so we had some questions about the latter part of your talk with the plot work. And we had one question or maybe you can clarify the invaded plots. Was there differences in number of plant species between the invaded plots and the not invaded plots? Would you mind just reviewing that for us?
Yeah, that's actually a really good question. Now I'm trying to think if we looked at the richness of each plot and compared that, and I think that maybe we did not. What we did do was to make sure that the number of plots across ecosystem types was fairly equal. So it's not like most of the invaded plots were forests
Mm.
or grasslands, and most of the native ones were something else. So that was fairly balanced, but richness, I don't know. And I can't remember. I don't even remember if we looked into that. That's a very good question. And I could see perhaps richness playing a role. Maybe in forcing invaders to be more competitive if richness is higher in an area. But although high richness can also indicate that a specific region is more productive, so it's just better for all plants to subsist there. There's just more resources. That's a really good question that I wish I could go now, like check really quickly on my computer so I can see what the breakdown across invasive versus uninvasive pods is in terms of richness. That's a good question.
Great, thank you. We had one question. This may not be something that fell within your study, but how much difference was there between the microbiome of the remnant or uninvaded versus the restored habitats? Was this indicated in the plots? So there was a simplification or a homogenization in the microbiome community, but can you speak to that difference a little bit more for us?
So if I understood correctly, I'm guessing this question focuses more on the actual what those differences were in the composition, if it was more bacteria dominated versus fungi dominated or something like that. And that's why I said that we don't know. We did not characterize these microbial communities that deeply because we were limited to that PLFA information. It was really hard to find microbial composition information and vegetation surveys for the same plot. So we were limited due to the data, but that's our plan for future research. Writing proposals and stuff, to be able to actually go into these plots, not just use this broader scale or broader resolution data that we have, get more specific into each plots and hopefully get that same magnitude or that same number of plots, and probably not, but so in other words, we kind of want to recreate this work or dig deeper in this work with finer resolution data so that I can be able to describe what's actually happening to those microbial communities in the future.
Great, thank you. We, a related question. So this research, do you think it's telling a story about the roots determining the microbial community, or do you think it's that the microbes are determining what plants can grow? Can you speak to that?
I love that question. Thank you so much. It was something I wanted to test this time around, and the data, we were limited by the data, and we couldn't. But I certainly have that question. Are the invasive plants changing the microbial communities? And is that then bringing more invasive plants, or is it the other way around? Is there some sort of positive feedback cycle going in there that invasive plants change the microbes a little bit, and then that just makes the environment a lot better, the soil more hospitable for other invaders? It's a fantastic question. Again, that I'm sorry I don't have that answer, but hopefully I will in the coming years, because I think it's a very, very, very interesting thing that could be happening, process that could be happening there. Kind of a mechanism of invasional meltdown. Very cool.
Definitely, thank you. Yeah, that is a really interesting, yeah, how cool. We had one question about, we use the term invasive versus, and sometimes maybe aggressive. Can you speak to the differences between an invasive plant and maybe an aggressive native plant?
I'm going to be a little bit facetious right now and say, can anyone
Right.
really do that? I think that that is one of the issues with just slapping a label like invasive or any of the other labels into the plant, that they are human created, right? So each person, depending on their context, professional context, their geographic context, even their language, that linguistic context, you might use a different term for these species. And that's why we're trying to break it down into either dimensions or something that is more ecological and more consistent to the processes that these species are engaging in. Now, in my understanding, I can tell you what I think is the difference, but it's not going to be anyone else's definition. Is that an aggressive species is native, but it behaves like an invasive species. And sometimes native species are invasive elsewhere. So that's something to keep in mind, right? Invasion also has to do a bit about your geographical context, because it doesn't occur naturally here, right? But our native plants might occur now and not occur elsewhere where they're not originally from. So yeah, so that's my understanding, that an aggressive plant or a weedy plant is native but behaves, it can be very dominant. It maybe has earlier phenology, so comes out earlier in the season, takes over. I know goldenrod is one of those species. And invasives, at least in the federal definition, is a species that does not actually or naturally occur in an area. They were brought into an area through anthropogenic vectors, and that could be as a soil contaminant, as a horticultural escapee. So that is, in my opinion, the difference, but other folks might tell you something else.
Great, thank you so much. We have one question. Can you speak to ways to prevent invasions? I mean, I know that you're looking at kind of which ones will come down the line, but have you considered what prevention strategies are available to us with this?
Yeah, I think that the suggestions I'll give will probably be quite superficial because it is not my area of expertise. But I would say that at least ecologically, um, taking care of your native communities first. So making sure that the native plants in whatever this area is are healthy and able to compete. That for one. For two, also higher diversity. There's some evidence, sometimes inconsistent, but that areas that have higher plant richness also are also less invaded. So those are two things, especially when we're talking about backyards and stuff like that. Having just a monoculture of a grass might not be as resistant to invasive than a native yard that is quite diverse and has different types of plants that play different functions in the plant community. That helps to, it gives some resistance. Biotic resistance is something that I study, but not really in that tangible. Well, if I'm out of my yard, what should I do? But ecologically, that's what I can say that will help.
Definitely. Thank you. Yeah, one of the strengths of, yeah, a really locked in, stable community. Yeah. So thank you for touching on that. And we have one more question, and then I'll wrap it up for today. And if there's other questions in the chat that weren't addressed, we'll maybe write them up in our email tomorrow. And we'll as well share your website and how to find your publications and contact and everything with everyone tomorrow. But the last question, again, this sort of community change with microbial invasion. Do you have any, so the data that you looked at did you have any pre-invasion data, or was it just kind of the moment after invasion or the one time point? Can you speak to that?
Yeah. We did have some plots that went back and forth, either from moderately invaded to highly invaded, and those were considered separately, those two different time points. So each time point for the plot is considered separately, while taking into account that it was the same plot. But maybe I should speak to some of the other research happening in my lab. I have a student who right now is wrapping up a project looking at what happens once a specific invader comes into a plot. So he's looking at reed canary grass and wetlands and garlic mustard and forests. So going from not invaded to now reed canary grass or garlic mustard is here. And then what happens after when it comes to are more invasives coming in or not? And he's found that when it comes to reed canary grass, once reed canary grass gets there, actually invasive richness goes down. So reed canary, and you can imagine reed canary grass just takes over everything. So native richness and invasive richness goes down. So fewer invasives. But the opposite was observed with garlic mustard and forests.
So if there is garlic mustard in forest, there can still be a richness of other.
Yeah, as the cover of garlic mustard increased, so did invasive richness.
Yeah, it's so interesting.
Mhm.
Wow. Well, thank you so much, Dr. Nunez-Mir. It's been a really great talk, and we've had great questions and great engagement. So thank you for your time again.
Absolutely. Thank you so much for having me.
All right. Thank you, Gabby and Lily, for guiding us through the live questions. And thank you to all of you who tuned in live for contributing to the conversation. Access to this recording and other related information will be sent to all who registered in a follow-up email tomorrow. Before we conclude today's webinar, I have a couple announcements about upcoming in-person and online events that you won't want to miss. First, our online programming. Register now and join us Wednesday, February 12th at 4 p.m. for our Grow Native webinar, Resilient Yards, Keys for Successful Water Management and Pollinator Conservation with Dan Shaw. Then register and join us on Wednesday, February 26th at 4 p.m. for our MPF webinar, Horizon 2, 1 part wildlife, 1 part ecosystem services, and 1 part renewable energy with Steve Mowry and Ben Kruger. For our in-person offering, I'd like to remind all of those tuning in, especially those in the St. Louis Metro East area, to register for our Southwest Illinois workshop, Stormwater Challenges and Native Plant Solutions, which is happening Friday, February 21st in Edwardsville, Illinois. The deadline to register is next Friday, February 7th. This workshop is for anybody looking for ways to be involved in the green community efforts in the St. Louis Metro East area, with a special emphasis on stormwater management. Get to know Grow Native professional members and partners, some of which will be tabling information and ongoing opportunities at the event. Registration, including breakfast, is $25 for MPF members and Grow Native professional members, $15 for students with a valid student ID, and $30 for non members. Registration for all of the aforementioned events and much more can be found at our website under the Events tab. Thank you so much for joining us. Everyone, have a great